Hello, dear students. In this lecture, we will discuss direct methods of gene transfer. We have already discussed indirect method of gene transfer via agrobacterium mediated transformation in previous two lectures. Please refer to different books and different web sources to study this, and we will briefly discuss some of the methods of gene transfer which are directly done. So, as you can see in the flow chart, the methods of gene transfer are of two kinds, indirect and direct. And indirect method of gene transfer is through a mediator or a vector, which is agrobacterium. So this we have already discussed. Please refer to previous two lectures. In this lecture, we will discuss the direct methods of gene transfer, and we will focus on microprojectile bombardment, electroporation, microinjection, and liposome mediated transformation. Also, we will discuss PEG mediated gene transfer in this. We will not discuss silicon carbide method. So as you can see here, these are some of the pictures of direct methods of gene transfer. So you can see that the first picture shows PEG mediated, second is for electroporation, then the next picture is of gene gun method of gene transfer, which is also called particle bombardment. Then next is your liposome mediated gene transfer, and last picture is your microinjection method. So let us discuss these methods one by one. So in PEG mediated gene transfer, the primary requirement is protoplasts. So the protoplasts are taken out, and then the gene transfer happens in the presence of a hydrophilic agent, which is called PEG. So PEG is polyethylene glycol. So here you can see in the first picture that protoplasts are taken, and then they are mixed with the DNA in the presence of PEG, and then transformation takes place in protoplasts. In the next picture, you can see that they have compared the transformation method via agrobacterium and then PEG method. So both results in the formation of transgenic plants. The only difference is that agrobacterium is indirect method and PEG is direct method, because we are directly taking the DNA or gene, and we are putting in the host cell. So uh, in direct methods of DNA uptake, we require protoplasts, and before these plant cells, which takes up the plasmid DNA from the medium, their cell wall is removed with the help of enzymes like pectinase and cellulase, so that protoplasts are obtained, and then the uptake of plasmid DNA molecules by these isolated protoplasts from the medium has occurs via many methods. So first method is PEG mediated DNA uptake, where the PEG, which is your polyethylene glycol, is used. So PEG is a thick organic polymer, which is a chemical agent which acts on both the DNA structure, which is negatively charged, and plasma membrane of the host cell. So the PEG actually stimulates the transfer of DNA in the plasma membrane of the protoplast, because it stimulates the process of transformation. Addition of PEG in combination with calcium chloride in the medium has been found to stimulate the process of DNA uptake, as well as the integration in isolated protoplasts. So the PEG CaCl2 stimulated DNA uptake, is reported in many plants, especially in monocots like Triticum monococcum and rice Oryza sativa, also in some dicot crops like oilseed, rape, and tobacco. So, it this was the method of PEG-mediated gene transfer. Now, electroporation is the second method where we directly transfer the DNA in protoplasts, and in this technique, the isolated protoplasts are given a shock treatment so that the pores are formed in protoplasts. As you can see that electroporation is made of two words, electric plus pores. So electric high voltage electric pulses are given to protoplasts, and which makes transient pores in the cell membrane, and because of these pores the plasmid DNA molecules from the culture medium passes through these pores in these protoplasts, and then after some time the cell wall grows back within a day or two, and the cell can again divide. So here in this picture you can see that we have a yeast cell, and then plasmids are there. So electric pulses are given, and then this plasmid is going inside. So here they have shown yeast cell, but we can also take out plant cell, and after giving electric pulses, when the pores are formed, the DNA goes inside, and these pores are then they seal themselves after a day or two. So at times only one, but the frequency rate of this transformation is very low, and sometimes only 1 in 10,000 or 1 in 100,000 protoplasts subsequently divide. So, and most of these cells do not form plants. That is why this method of gene transfer is not very widely used compared to agrobacterium mediated transformation. So when you see, this is the method of electroporation. Here you can see that the protoplasts are taken, high pulse electrovolt is given, which is resulting in the formation of pores, and through which the DNA is going out, going inside. So here you can see that uh, electroporation, three pictures are shown here, and all the pictures are showing that pores are formed in the presence of electric pulse through which the DNA is going inside. The next method is liposome mediated DNA uptake. So in this method, a suspension of positively charged liposomes, which are vesicles of oil or fat, is mixed with the negatively charged DNA molecules. And why this is done? It is to wrap up the negatively charged DNA molecules or genes into liposomes so that they can be mixed with the DNA. So, and they can be transferred in the protoplast. So here you can see in the picture that DNA is there, DNA is negatively charged, liposomes are positively charged. So lipoprotein complex is formed, and then it is going inside the plasma membrane of the protoplast via endocytosis, and this is how this goes inside. So this is also not widely used when you compare it with agrobacterium mediated transformation. So this is again the same picture. Now next method is your micro injecting the DNA, which is done with the help of micro injection or a macro injection. So here uh, this is a method of inserting DNA into intact plant cells, and for this the protoplasts are not required. So the DNA can be injected directly into the cells, and or individual cells. So in the, what in this technique, This technique requires an elaborate apparatus which consists of a microscope and delicate micromanipulators to hold the cell, view the cell, and then inject DNA into them in a solution containing DNA with fine glass hollow needles. But the microinjection method is also not very popular because of the cumbersome technique, and the frequency is very low when you compare it with agrobacterium mediated transformation. So here in this picture you can see that there is a pipette for holding the cell staple, and there is an injection which contains the desired DNA. So when the diameter of the needle is less, we call it micro injection. When it is more, we call it macro injection. So here you can see the pictures on different pictures and different steps involved in micro injection or macro injection, and how it is going inside. So in A you can see that these are this is a cell, and the it is holded. Then in B the needle is going inside with an injection needle. The DNA is transferred, and in last picture you can see that successful injection with swollen pronucleus. This is again some more pictures. Now the last method we will discuss is microprojectile bombardment method, which is also called ballistics or gene gun method. So when you compare it with other methods of gene transformation, microprojectile bombardment or ballistics, uh, the uh, the credit for this goes to J.C. Sanford and T.M. Klein, who in 1987 constructed a microprojectile gun or DNA particle gun, which is marketed by DuPont, and it is used to deliver DNA directly into plant cells by shooting it through cell wall and cell membrane. So this microprojectile gun, it results in the formation of holes in the membrane which rapidly closes by themselves. So in this technique, these microscopic particles of gold and tungsten are coated with plasmid DNA molecules, and then they are fired into target cells, which are your plant cells. So, in a proportion of cells, the DNA is transferred to the nucleus, and it results in the integration of the transgene into plant genome, which is then passed on to the cell's progeny. So the tungsten or the gold particles are very small to penetrate the individual cells without destroying their integrity and viability. So one of the earliest and probably the most commonly used is the ballistic particle accelerator, which initially employed a standard .22 caliber gun cartridge for firing the microprojectile, which then travels down the barrel and hits back of stopper plate. So the DNA coated pellets then pass through a central hole in the stopper plate into plant cell tissue placed below. So let us see some pictures. So this is the picture of microprojectile gun, and you can see that the details that um, it is going via gold and tungsten particles. The DNA is in the form of bullets, and this is the microprojectile gun. And this is the picture of a microprojectile gun or particle gun, which was developed by C. Sanford and T.M. Klein. So this is the summary of this method. And the advantage of this method is that many species which are recalcitrant to other direct transfer methods and which are not readily transformed via agrobacterium mediated transformation can be used, can be transformed via this technique. But the major limitation is that the frequency of transformation is very low when you compare it with agrobacterium mediated transformation. So, I think students, we have discussed all the techniques of gene transformation via direct methods. And thank you, students. Let us meet in next lecture.